Welcome to this explainer. Okay, I want you to imagine just for a second that your entire body, your skin, your brain, your heart, your muscles, literally the whole shebang was originally built from just one single tiny cell. Kind of sounds like pure science fiction, doesn't it? But honestly, it is absolute biological fact. So today, we're going on this truly fascinating journey right into the microscopic world that builds our bodies. We're going to decode the mystery of multiplication to figure out exactly how life scales up from the microscopic to the massive.
Okay, let's dive into this and map out our journey. We'll start with the miracle of multiplication, head into the lab for the onion root experiment, uncover preparing for division, and tackle mitosis versus meiosis. Then we'll discuss what happens when division loses control and wrap it all up with the foundations of life.
Section 1. The miracle of multiplication. Now, before we look at the mechanics, we really have to set up this profound biological problem we're trying to solve today. Namely, how does one single cell become an entire body? It's a pretty mind-bending mystery when you stop to think about it. If you start out as just one single biological building block, what exactly is the mechanism that allows that block to copy itself over and over and over again to eventually construct a fully functioning human being? And going beyond just building the body, how does nature actually repair us when we get cut or replace old cells that have simply worn out?
Well, the scale of this operation is, frankly, staggering. Trillions. That is the actual number of cells operating inside of you right now. Trillions of cells all originating from that one initial cell, dividing and multiplying relentlessly. It's really nature's ultimate way of renewing and building itself. But to actually understand how a human cell divides, scientists ran into a massive problem early on. You see, dividing cells are highly transparent, which makes them incredibly difficult to see live. They desperately needed a window into this invisible microscopic world.
Which brings us to Section 2, the onion root experiment.
To crack the code of human cell division, researchers first had to find a reliable way to make this invisible process visible. And surprisingly, they found their perfect biological window sitting right in the kitchen, the humble onion. Or more specifically, the onion root tip. The very bottom of a growing onion root is this highly specialized growth zone. The cells down there don't just sit around chilling, they divide extremely fast to push the root deeper into the soil. Because this area is an absolute hotspot of rapid, non-stop cell multiplication, it gives us the very best chance of catching cells right in the middle of the act of dividing.
So how do we actually pull off this experiment? The lab steps are super precise and totally fascinating. First up, you place an onion bulb over a water jar and just let those roots grow for about 5 to 6 days. Once you have fresh roots, you snip a tiny 2 to 3 centimeter piece from the very tip. Now for the crucial chemistry step. You add dilute hydrochloric acid. You absolutely need this acid to soften up the tough plant tissues so the cells can be separated. But remember, the chromosomes inside the cell are practically invisible, so you add a stain called acidocarmine. This brilliant dye swoops in, binds to the cell components, and turns them a vibrant red or pink color. Finally, you gently warm the slide and squash the root tip totally flat under a glass cover slip. This physically spreads the cells out into a single layer so the microscope light can shine right through them. And this brilliantly illustrates exactly what's going on. When you look at that squashed, stained onion root under a microscope, you can actually see the different stages of cell division completely frozen in time. You'll see cells where the newly stained pink chromosomes have arranged themselves into a perfectly clear line right down the middle. In other cells, you catch the exact moment those chromosomes physically pull apart, migrating to opposite ends of the cell. Finally, you can observe the cytoplasm itself physically dividing right down the center, which leaves you with 2 distinct newly formed nuclei. It is literal, visual proof of how a cell splits to drive our growth and repair our damage.
Moving on to Section 3, preparing for division.
Seeing that physical split happen is totally amazing, but we kind of need to shift our focus to the hidden, incredibly time-consuming preparation that makes that split possible in the first place. You see, a cell doesn't just suddenly decide to divide out of nowhere. In fact, the actual physical division part, that only accounts for about 5% of a cell's entire life cycle. The other 95% is purely preparation. Think of it kind of like a mother cooking a massive Thanksgiving dinner for her family. She might spend several hours in the kitchen meticulously prepping the ingredients, chopping vegetables, and cooking the meal. But then it only takes the family about 5 to 10 minutes to actually sit down and devour the whole thing. A cell works the exact same way. It spends 95% of its time just growing and meticulously copying its DNA, all so that when the time comes to divide, that 5% execution phase happens absolutely flawlessly.
And that DNA preparation, that is the ultimate master control mechanism.
To prove just how unbelievably powerful DNA really is, scientists back in 2010, led by Craig Venter, pulled off this totally awe-inspiring experiment. They sequenced the DNA of a Mycoplasma bacteria and then literally synthesized an artificial, lab-made version of that DNA using basic chemicals. They removed the natural DNA from a recipient bacterial cell while keeping its cell membrane and cytoplasm intact and inserted their artificial DNA. Amazingly, the cell accepted it and just began to normally grow and divide. This definitively proved that DNA controls a cell's structure and activity. Now, it's definitely important to note here that they didn't create life completely from scratch. They still needed the host cell's natural cytoplasm and membrane. Creating an entirely living cell from absolute scratch, well, that remains one of biology's most beautiful scientific mysteries.
Section 4. Mitosis versus meiosis.
Okay, so we know how cells prepare to divide, but they don't all divide for the same reason. So the crucial point is understanding the stark contrast between the two main mechanisms. The first is mitosis. Mitosis is all about replication and identical cloning. One single cell divides one time to create two completely identical daughter cells, maintaining the exact same number of chromosomes. This is the everyday engine for your body's growth, healing cuts, and replacing old cells. But then we have meiosis, and meiosis is an entirely different beast. Here, a single cell actually divides twice to create 4 genetically unique cells. Most importantly, these cells only contain exactly half the normal number of chromosomes. Why half? Because meiosis is exclusively used for reproduction.
Let's quickly break down how meiosis works in animals, humans included. This process creates our gametes, the sperm and the egg cells. Because meiosis strictly halves the chromosomes, male cells, which naturally have X and Y chromosomes, will produce sperm that carry either an X chromosome or a Y chromosome. Female cells, which are XX, produce eggs that only ever carry an X chromosome. This leads to an incredibly fascinating biological fact. Because the mother always provides an X, it is actually the male's sperm, whether it brings an X for a female baby or a Y for a male baby, that ultimately determines the sex of the offspring. And just a quick side note, this isn't exclusively an animal thing. Plants totally use meiosis too, creating pollen in their anthers and eggs in their ovaries.
Because meiosis involves mixing up genes and strictly having chromosomes to be paired up with a partner later, it acts as the fundamental engine of biological diversity. The completely unique genetic mix inside every single sperm and every single egg ensures that no two individuals on Earth are exactly alike. Without the variation created by meiosis, human diversity as we know it simply wouldn't exist. It is literally the reason we are all beautifully unique.
Section 5. When division loses control.
We've seen how perfectly orchestrated this microscopic mechanism usually is, but we absolutely have to look at what happens when this delicate system makes a mistake, because it is literally a matter of human health and survival. When the careful rules of mitosis or meiosis are broken, the consequences can be severe. If mitosis loses its regulatory brakes, a single cell might begin dividing completely out of control. Instead of stopping when the body has enough cells, it just keeps multiplying, eventually piling up to form tumors. On the reproductive side, if meiosis makes an error and the chromosome numbers don't halve properly, or if genes are improperly copied, it leads to genetic disorders. This can cause significant developmental problems and physical differences in a baby, and it can also be the underlying cause of early pregnancy loss or reduced fertility. The perfection of the cell cycle is quite literally what keeps us healthy, and its failure is at the root of our most serious diseases.
Section 6. The Foundations of Life.
Let's move to and see how this builds. Let's zoom all the way out from the chemical mechanics and the physical mistakes to look at the grand historical theory that ties all living things on this planet together. Back in 1838, a German botanist named Matthias Schleiden made a groundbreaking report. He realized that all plants, no matter their shape or size, are fundamentally made up of cells. Just 1 year later, in 1839, a German zoologist discovered the exact same thing was true for all animals. These were massive discoveries that laid the groundwork, but it took another scientist, Rudolf Virchow, to really expand this into the classical cell theory as we know it today.
Virchow looked at all this evidence and came to one monumental conclusion, beautifully summarized in this incredible quote. All cells arise from pre-existing cells. This single sentence connects bacteria, fungi, plants, animals, and you into one unbroken chain of cellular life. A cell doesn't just magically appear out of thin air. It has to come from a parent cell dividing. Every living organism is built on this basic functional unit.
So, as we wrap up this explainer, I want you to literally look down at your hand right now. Take a second and think about what is happening under your skin at this exact second. Can you feel the trillions of individual cells working, copying DNA, and preparing to divide, all just to keep you alive and functioning? As you go about your day, ask yourself, what other microscopic miracles are happening inside you right now, completely undetected? Keep exploring, keep questioning, and I'll see you in the next explainer.